Next piece I'm going to talk about is called variant quality recalibration. Again, this is another level of recalibration. So, and this is recalibrating the variant quality. So we use the SAM tools and a few other slides that before this, we talk about called variant, and then we always have a quality associated to that. But how accurate that quality is a question, and it becomes a not easy question. Okay, and similar as the base quality report, variant quality is not always accurate as well. And the problem here is once the base quality is not accurate, we have someone to blame. We blame the Illumina or solid. But when variant quality is not accurate, we only can blame ourselves. So it's because of the mathematics equations are not perfectly right, or alignment is not right, and things like that. So after variant recalibration identification, we will identify a lot of variants. How many of them? I will give you a number, and there's a good slide on that. And I just got the numbers today, so not generated by me. I Googled and they showed up. So we'll go through that. But many of these variants are real, but a lot of others are not. Especially this type of thing, the more thoughtful you are in the previous analysis, the less error we will identify here. But still, there will be a variance here. But for some of the methods, you just throw it in some of alignment algorithm, throw that in some variation identification. And if that's the case, there will be a lot of, I think this is that. There will be a lot of miscalibration there. So there's a similar as the basic errors. There is a lot of factors that determine this. The first one is the machine chemistry artifacts. This includes sequencing cycles, again, different chemistry versions. You probably don't know what that is, but the people who run the core knows it. And these manufacturers, they say, let's try this kit. Let's try that kit, and give you different results. Okay? GC-rich regions are harder to sequence. The homopolymers, the sequencing files, so on and so forth. There's a lot of considerations. So, but those are the machine or chemistry artifacts. There's the analysis artifacts as well. For example, the systematic alignment inaccuracy, or you use the wrong variant call algorithm to determine the variance. Those are all causing troubles. Each variant has many variables. Okay, there's a variation quality so that we can calculate, and also there's the depth of coverage, a strand bias. We will go through that a little bit in later slides. There's so many things that associate to that. This is one of the figures that I got from internet, which is a GATK package, one of their presentations that they made to the public. So you can see that there's two distributions in each figure, and this one represents the ones that, let me see, the known variations, meaning the variations is already documented in dbSNP 132. So those are most likely to be the real variants. And the red one here are the ones that are not documented in the dbSNP. It's not mean they are wrong. Okay, it doesn't mean they are wrong. dbSNP is not really complete, and a lot of variants we identified are rare variants. So it's not going to be in there. But a lot of them are errors as well. So this is a mixture of, most likely it's a mixture of errors of all the novel variants. So you can see that for the variant quality distribution, this for the ones that are already known, it follows this bimodal peaks. For the ones that are not, it follows this shape. You can see the quality of variant difference, and clearly that the variant is already known, have a little bit higher quality. But this is for each individual variant, and we can really calculate this variant quality. And there's a haplotype scores, and these two are probably about similar. Extreme Hardy-Vandenberg violations, I don't have time to go through the genetics base of it, but but you can see there's a difference between the good ones or a mixture of good and bad ones. All right. So this is a strand bias, and you can see. I think beyond this point, there's more bias absorbed, and there's the differences as well. This is, by the way, this is one of the studies, right? Depths of the coverage, they are the same, and the root mean square, that's what RMS means, root mean square of mapping quality. A lot of mapping qualities, we just have a summarized number for that. They are pretty similar. A mapping quality rank sum test, and this is a read position rank test. But the point here is there is a lot of different variables can be associated to each variant we identified. And how many of those? And there's you can get your own list as well. With different population, the good SNPs or a mixture of good and bad SNPs, their distributions are slightly different. Okay. Actually, most of the information that we need are in the VCF files. Remember, you guys were all complaining that I go through those boring VCF files, but turns out that they have some useful information. So all these informations, I don't remember the code. What the DP mostly snaps 87 and so but some of them I don't. And so strand bias and things like that. So they all have already pre-calculated values in these VCF files. That's why I want informatics students to go through those files to get your hands dirty, because there's many information are there. And keep in mind that this is just for one variant. We have so many information in these VCF files. Question here is, which variants are more real? Okay. The common strategy here is we are using, before we do this variation quality recalibration, because that paper was only published this year, right? Before that, we don't have this recalibration procedure. So the common strategy that most people use is use a different cutoff on each variable. For example, I only trust the variant with quality value larger than 20, which means the p-value is smaller than 0.01. Okay. For example, the strand bias is less than 5%. For example, the depth coverage is more than 8. For example, the root mean square of mapping quality is larger than 20. For example, many things. So for each individual variable associated to this variant, in theory, you can make your own cutoff. But once I make one or two decisions about the cutoff, I feel okay. But once I have to make so many cutoffs, I'm feeling I'm a little bit too arbitrary. If there's anything wrong, there's too many parameters, it's too arbitrary. So a better approach will be based on all these variables. How can we assign a well-calibrated probability, one continuous value, and to each variant call in the call set, one can then create highly accurate call sets by filtering based on these single estimates. I still need to make arbitrary cutoff, but I only need to make one arbitrary cutoff rather than 20 of those, which is always too scary to me. Does that make sense, the rationale behind this? Okay. So the recalibration strategy is training on highly confident known sites to determine the probability that other sites are true. Okay? And computer scientists call it machine learning. Or statisticians call it regression and then recalculate the probability or things like that. But anyways, regardless how, so here is one example of a machine learning strategy. So for each individual variant, you will have many different variables. So we just mentioned the quality, mapping quality, the sequencing depths, or so on and so forth. There's so many things. And then we can have a confidence known size, which we call the gold standard. So those sites we know it's more likely to be correct. How do we know that? The strategy is to pick the ones that is already documented in the DB SNP. If it's already documented there, it's more likely it's the real ones. Okay. Again, that doesn't mean the remaining ones are not real. But in the DB-SNP, and this is more credible. And then the computer scientists will train a machine to learn the patterns. And for me, what machine learning means is so usually my way to do that is I would still first go to statistical approach and trying to get a distribution or equations and things like that. And every time I failed, I'm not smart enough to get out the equations and to do it, I would just try machine learning approach. And then I would just use the neural network. Here are the things, throw that in. Whatever the outcome is, I would take it. And of course, you have ways to evaluate that, right? But anyways, so basically, you base on a lot of gold standard with the variables and train a machine. And then for any other variance, and you will let the machine to decide whether it's a good one or bad one. So this is the basic procedure for the recalibration. Does that make sense? All right, so it's simple. The gold standard in this approach they're using is annotated variance, which is identified variance already present in HapMap 3. Okay. Again, HapMap is an international project. We probably didn't talk about too much. They got, it's many years ago, they have HapMap 1, phase 1, and phase 2. I think they initially they did genotyping for 100-something individuals, and with four populations, I think. And then they extended the phase 3 include 1,300 individuals, and they did whole genome sequencing, which is part of the 1,000 Genome Project, right? So the gold standard they are using here is the annotated variance that is already identified in HapMap samples. And the models they use is something called a variational Bayesian Gaussian mixture model. So every time you got so many confusing, doesn't make any sense, the terminologies connected together, let's just consider it's okay. So I will consider this is okay, right? I'm not going to go through that. So let's see. But anyways, let's go through this. Probably not. But anyways, these are the equations. It looks pretty confusing, but it's kind of very similar to regression type. So when statisticians look at this, you guys just go ahead to get the paper. It takes, I promise, it won't take you more than 5 minutes to figure this out. Okay, I just don't think this is the right place I talk about this. And also you can use some other tools, right? You can use supporting vector machine or neural network and type of other machine learning type of tool to improve this thing. But this happens to be the way that they are doing it. Using those equation parameters, variables, train the machines, and get a new variant, and let the machine to decide what is good, what is bad. So here is some of the results they got, and this is for the training phase. So you can see that this is two variables here. X here is the variant quality score. Okay, so based on SAM tools, I can calculate variant quality. The more towards the right, that means that variant quality is higher. And the Y here actually is the strand bias evaluation. So this is the lower is less bias, the higher is more bias. Okay, so the data in the HapMap samples really look like this. And the red ones are the SNPs found at HapMap 3 project, and the yellow ones are in DB-SNP. DB-SNP, we know there's a lot of junks in there. And they really have these two bigger clusters, and this one representing the heterozygous variants, and this cluster represents homozygous variants. And there will be some equations associated to that. But the point is, when they use the new sample, a new variant, they sequence the sample and they go through all these hassles. Eventually, for each variant, they calculate a variant quality and all the other variables. And then they put that into these machine parameters, and they found out these things. So these regions, most likely, these are the score are lower, and together with the bias are higher. The sequence bias are higher. The strand bias are higher. These are probably the noise. So this is retained. The green ones are the retained novel SNPs, and the purple ones are the filtered out novel SNPs. So based on this simple strategy, I know equations looks more difficult, but that's what statisticians do. Right? They just want to use those equations to confuse us. But bottom line is the principle is simple. You train the machines, you get a bunch of parameters, you put your new SNPs in, and you can figure out which one is potentially more real than others. The variant recalibration successfully increased the TI-TV ratios for the confirmed variants. So do we still remember what is TI-TV ratio? We'll get into that a little bit, but I talked about it last time, if I remember correctly. And this seemed to work well, and variables can be added into the original list as well. So what I mean here is in the GATK package, they have this variant recalibration functions. They already have a lot of variables built in. Variables meaning it's sequencing depth, strand bias, variant quality, and mapping quality, and the things like that. But if you think there's a need that you want to put in more variables in this, and you can certainly do that, and that is allowed, and it's perfectly okay. So this is not a separate topic, although I listed it in this way. So I mentioned this just conceptually went through this variant quality recalibration. And then for the novel variants, we have some retained variants, which are good variants, and the filtered out variants, or bad variants. But the point here is, how do we evaluate a SNP quality? How do we know that after this type of filtering, these difficult equations, and the results actually improved the SNP quality? Well, they retained all the good ones and they threw out all the bad ones. How do we know that? So actually, there's a couple of strategies or measurements on the SNP quality evaluation. The first one is expected number of calls. This is simple. It won't give you any quantitative information, but it's just conceptually, if for one individual you identified 100 million variations, that's most likely your sample is contaminated with mouse, and it's not going to be human, or something is wrong with it. So you want to have a general ballpark where your data lies. And the number of SNP calls should be close to the average human heterozygosity of one variant per 1,000 bases. Okay? Here is some of the equations, and the equation number of polymorphic sites you want to expect. And L here is the length of the regions you sequenced. And theta is something they call it, where is it? Heterozygosity. Actually, it's different for different populations. I don't know enough to comment on this, but I assume this assumes how diverse the population is. Probably, for example, Caucasian population probably it's more uniform compared to African populations. That's my assumption. I just assume it that way. I don't know whether it's right. But anyways, these are a parameter for each population specific. So the information I want you to take a look at here is for the European population, for the whole genome sequencing, you are expecting about 3.3 million variations identified. Okay? If you identify 100 million, there's something wrong. For the exon sequencing, for the 32 megabase exon sequencing, that is representing the total number of race, the region of exon you are expecting about 20,000. And for some of the data that we have, and it's a broader capture array, it's a 50 million base extreme exon regions, and we generally look at 38,000 to 40,000 variants, which is in the ballpark. For the African genome, the whole genome, you can see that we are expecting 1 million more variants comparing to the European populations. And so here are the samples. But these are really for one individual, okay? These are for one individual. But when you talk about 100 samples together, and these are the numbers you're expecting, 100 European people, you may find about 13 million variants. Anyways, I just thought this is not really off the context, but not very closely related, but I think this is a very, very important information that once you identify the variants, you want to go through it and whether your data really makes sense. And the second type of evaluation is the concordance with the genotype chip counts. Okay, that means that for some of the samples that initially they already did genotyping arrays, okay, unselected variants, and you want to know that how much that your new sequencing data agree with. You may think, well, if we already have arrays, how likely that we will have arrays and also have the exon sequencing data? Well, actually, it's pretty likely now because the array price is dropping as well, and some of the studies they do both. They do both. So we do have some samples. Studies, actually hundreds of samples, has both genotyping arrays and together with exon sequencing. And this really helps us to do the quality control at the very least. Of course, we use that information to do something else as well. And another type of evaluation will be what fraction of SNP are already known. Okay, if you identify exon sequencing 40,000 variants, and how many of them are already known, are already documented in dbSNP. So dbSNP calculates most common variants, so most of the true variants found will be in dbSNP. For single sample counts, usually 90% of the variants should be in dbSNP. Okay, again, this is nothing concrete, but this is something that you want to expect. And need to adjust the expectation when considering costs across different samples. And the next evaluation, whether this is a good SNP or bad SNP, will be so-called transition to transversion ratio. Okay, I remember very clearly that we had one slide on that. So what is the transition? Transition are the changes that within purine or within permeating. And the transversion are the changes that between purine and permeating. Okay, and transitions are twice as frequent as transversions for the real biological signals. Validated human SNP data suggests that the transition-transversion ratio should be about 2.1 in the genome-wide and 2.8 in exons. Okay, and what is that? False positive SNPs should have. So false positive SNP by random, the transition transversion ratio should be 0.5 because we have only one choice versus two choices. Okay, so transversion, this ratio is a good metric for assessing SNP qualities, and we have seen that after the recalibration stage, that after this recalibration, they really improve this transition transversion ratio pretty dramatically, and which is a good comment. Are we okay? Are we still okay? Okay, we are 25 minutes away from the end, but I'm going to use up this time. Okay, hanging on here. Let's talk about structure variations. Okay, I worked until 3 o'clock this morning to work on these slides, so you guys just need to be patient and come stay with me. I have to go through it, okay? And for the structure variations, we already talked about there's a lot of different type of structure variations. They are traditionally defined as deletions. This was one of the old slides. And those deletions, insertions, or inversions larger than 1 kb. And they are often involved in repetitive regions of genome and complex rearrangements. And there's no optimal methods for structure variation discovery before next generation sequencing. There are ways to do that for the copy number variation, but that's it. For the other type of translocation or things like that, there's no optimal way to do that. And structure variation will affect a larger fraction of the genome as compared with the SNPs. Okay, we know that because they represent thousands of base pairs, whereas SNP only represents 1. And this is important. Analysis of 1,000 genome project data identify a total of 1,700 structure variations that affect the coding sequences. This is stunning. And these are supposed to be healthy individuals that are in the 1,000 genome project. Okay? So this is a really, really important type of information. And actually, there's another one that you probably are more interested in because there's no good way to study structural variations. There's a lot of disease-causing ones are still hanging out there, was not discovered. Okay, if you want to find out what is causing disease, those are really good low-hanging fruits are there to be picked. There's many different types of structure variations. We talk about there's deletions, insertions, inversions, copy number variations, segmental duplications, or there's many different types, and there's some of them are really, really complicated. You cannot even draw those figures. Okay, now we come to the dirty part. Okay, so those are good and the information that we need to know. But here there's a summary of the methods based on tag density. So there's basically two types of method to use next generation sequencing technology to study structure variations. The first one is the method based on tag density, and which here is using the depth. The first one is a method using depth of coverage. Genome is divided into, usually the way to do it is divide the genome into multiple window size and count the number of reads that fell into each window. So this is more suitable for the copy number variations studies, okay? Which is straightforward. So you can see there's a duplication here, this is a deletion here. Again, think of our mappability issue as well, right? So this is perfectly valid. The assumption here is sequencing rates are randomly sampled with equal probability from any location on the sequenced genome. Thus, after aligning these rates to the reference genome, the rate density of a given genome window should be proportional to the local copy number. So there's duplication and deletions. And the density is also determined by other factors, including GC content, mappability, and things like that, right? So we know that. We just need to figure out a way to deal with it. Actually, there are some approaches. I put some of the references there, and how you will put them out and put it in the ENCORE system. And the first paper actually did some work on correcting these signals based on this GC content and mappability issues. So those has been considered. But anyways, this is really called the method using depth of coverage. And at this stage, you probably can think of, oh, it doesn't really matter whether this is a mate pair or pair end or single end approach. As long as I count the numbers, we're good. Okay? And the second type of strategy is still based on the tag density, is to use a case control type of design. So think about this is for each individual, I'm just comparing different genomic regions for this one. But this is for using the depth of coverage. But here, for the case control designs, actually I can compare the red ones are the case, are the disease sample, and the blue ones are the controls. So I don't really need to worry about those mappability or GC content because they are going to be the same for these case and control groups. I just compare in this region whether there's, so this one probably there's a copy gain in the disease sample, there's a copy loss in the case sample. This is kind of straightforward. Okay? And the assumption here is the bias for case and controls are similar. The bias meaning the GC content or the whatever is considered mappability and things like that. So the major application for this type of study is comparing tumor genome versus matched normal genomes. So we know that some patients that they have their, for example, get breast cancer and get the tumor samples, and also we can do the same thing for their blood samples, which is supposed not to be tumoric, and to compare that to see whether there are some more copy gain or copy loss in the tumor samples versus the normal samples. Okay, the strategy for this is the real algorithm usually is include the 3 different steps, which is really, really simple, straightforward. The first one is the partition, meaning we want to separate the genome into different window size, okay, into different windows. compare and tag density in each window. Just define the windows and count the number of arrays for into each window in the disease and the normal condition. And just the third one is, and the second one is to compare them, which regions are different. And the third one will be to correct for the multiple testing issue. Same thing as a microarray or anything, multiple hypothesis testing involved. But the choice for the window size using this type of strategy is not that straightforward, actually. It's kind of tricky. Because if your window size are too big, of course your signal will be really robust, but your resolution becomes very large. So your resolution is not very good. Okay? You want to figure out a small copy number variations, small regions with copy number variations. But if you define your window too big, you are messed up with your resolution. However, if you define it too small, that also decreases the power, because the more windows you defined, the more tests you have to correct. So once you come to the multiple hypothesis testing issue, you're decreasing your study power. So that is an issue. So the solution for this, there's a couple of solutions. So that's what I was very surprised, because this is basically not my research area. I'm learning here together with you guys. When I studied the references last night, looking at papers, this is really very active research directions. I just didn't realize. So the solution for this is, the first one is we can optimize the window size given a significant level, a log2 copy number threshold, and the coverage of the data, and then the window size, and then further do the window merging. So basically, you are deciding your window size based on the goal you want to achieve. And that is a very smart and good approach. And this was published in 2009. And another method was published also in the same year, is focus on identification of the breakpoints rather than copy number variation regions. Meaning, I don't do the predefined region. Okay, I first identify the breakpoints. I didn't know how they do that, but you can go to study. And then the regions can be big, can be small, and then you use those regions as a unit and to do the copy number identification. Very interesting, and we go to look at this paper. So this is a general strategy. These are the ones most likely will focus on the copy number variation or deletions. So because when you do this type of analysis, you are really based on the tag densities, the number of arrays that fall into each genomic locations. Does that make sense? Okay. And the second category of method is the methods using paired end mapping. Okay? So we went through this last time. So the general strategy here is to align the paired end reads from a sequenced genome onto a reference genome and look for discordant pair end mapping that may indicate the presence of structural variations. For example, if you see they are too close to each other, once this is the case, this is the reference, and once you map it, they are longer than you expected, there's a deletion in your sample. And the insertion, or then this you can see the translocation, so there's a connection of two chromosomes, and this is probably a deletion because the middle part they are not mapping it, or things like that. So there's the method to using pair end mapping. This is something really new. Only after you do the next generation sequencing technology, and this can be possible. Okay, just very briefly overview the pair end, because we are talking about it, and I'm sure you all know about it. For the students, we even did a quiz on this. So for the single end, just briefly overview. Single end, this is a 100 base pair, but only one end of the fragment. The fragment length is about this. And for the pair end, the same fragment length, but we are sequencing both two ends. And for the mate pairs, these are much longer regions, and we sequence both two ends. But what I'm talking about here is the pair ends. Okay? And in terms of methodology itself, there are two different types of methodologies. Again, two subcategories. The first one is the cluster-based methods. meaning that we'll classify the pair end mapping as a concordance and discordant pairs, and then cluster the ones that doesn't agree based on the positions of the pair end arrays. So basically, you just count it out. This part always having something pairing with another chromosome, and this is more likely to be a translocation and things like that, right? And there are a couple of methodologies has been designed to really help to identify this. But these are really for only the large structure variations. For some smaller ones, there's a distribution-based methods. So that is more as counting the distance between the two ends, okay, after you align it back to the genome. So one example shows here is, this is an example of a homozygous, is that insertion or deletion? I think this is a deletion. I need to get back to it. But the point is, this gray histogram is the read length that you are expecting, but after you map it to the reference genome, it becomes the length increased. That is a very good indication of there's a smaller deletions that in this region, so that once you map to the reference genome, it jumps and grows a little bit. And here is, if you look at the distribution look like this will be the heterozygous insertion or deletions. And so one is really similar to the what you expected, and which is the same as reference genome, and the other one has probably a couple of hundred base pair deletions. So this is a distribution-based methods, and it's a very nice study. So it's very smart. So we're done today. So next lecture, the part one, there are two parts. The part one will be Dr. Freud from our department. We'll talk about using exome sequencing to study the Parkinson's disease. And I will wrap up with the second part. I will finish the DNA sequencing part, which is on the prioritization of genetic variants, meaning that after you identify those variants, what are we going to do with it? How do we say which one has changed the amino acid sequences or changed the splicing regulation, and what are the informatics tools that are available there? Okay? All right, everyone have a nice weekend. We are not going to have a quiz.